During the during the first part of my presentation, I will be talking about back-to-back TOPO expression system, like mostly in general, and second part will be devoted exclusively to some of our results generated by using this system. Back-to-back TOPO expression system from Invitrogen is really very efficient, quick, and relatively easy way to generate recombinant baculoviruses for higher level of protein expressions. There are two major components of this system. The first one is they offer a choice of two different vectors. It's PFAST, both like they have the same background, and only difference between carboxy terminal or amino terminal location of epitope, which are used for purification purposes. And all this, both of these vectors, they allow rapid generation of an expression constructs containing your gene of interest under control of baculovirus specific promoter. And typically, system comes with chemical, like in this instance, it's one-shot chemically competent cells, but its cells type is irrelevant at this point because any competent cells can be used. Second important component of this system is very specific E. coli strain, host strain DH10 Bac, and this strain contains already like transformed baculovirus shuttle vector or BACMID. It's basically the entire baculovirus cloned inside of this plasmid. And it also contained a helper plasmid which encodes for specific enzyme transposase. And all this system combined allows generation of recombinant BACMIDs containing your gene of interest like for the next step of baculovirus construction. System also has like specific reagent for transfection. They do suggest to use sulfactin 2 reagent, but most likely some other reagents can be used as well, and also has some control plasmids which can be used for construction of control baculoviruses if something goes wrong with your system. Few words about some of these vectors. CT version of this PFAST Bac means we have six his epitope, which is used for purification, is located at carboxy terminus of your construct. Vector also contain TEV protease sites, which is designed primarily in order to allow, if for some reason you don't like the presence of his tag on the final product, you can always cut this tag by using TEV protease. And of course, if you have carboxy terminal location of his tag, you're supposed to include initiation codon on your gene of interest, and stop codon supposed to be removed in order to allow reading through this sequence. Next, amino terminal version is very similar. Basically, everything moved at the 5' end of the construct. And in this case, you do not supposed to contain your ATG initiation codon in your sequence, and termination codon has to be present in your gene of interest during primer construction. This system has several obvious advantages over a traditional method which is using homologous recombination. First of all, system allows blunt end cloning of PCR product. As a result, you can use enzyme like Taq polymerase, which has proofreading activity. As a result, you can avoid introduction of any unwanted mutation in your gene of interest during amplification step. And this system, at least theoretically, they I mean, they promise construction in less than 2 weeks, but it's very dependent from your hands. Let's say you can still spend much more time for this system, but it's definitely under equal condition. It's at least 2 or 3 times less time than was required for old system, which use traditional homologous recombination. And the most important part of this system, it's this system reduces the need for multiple rounds of plaque purification, because the main problem with the old system was if you have even smallest contamination of wild type virus typically grows much better than any recombinant. And as a result, after several rounds of amplification, all those wild type will take over, and as a result, instead of producing your own protein, you will be making infected cells by wild type virus, which basically empty vector without any protein production. And system also, because of convenience of working with E. coli, you can make at the same time a lot of different variants. It's relatively easy, and you can like produce multiple recombinant molecules at the same time. The main, I would say, disadvantage for this system, it's very steep price. For example, for the entire system, which allows construction of 20 different recombinant baculoviruses, cost close to 2,200. And the main problem with TOPO, as soon as you finish your vector, you cannot make by yourself. You have to buy a new one. In our case, in our lab, we started, because this TOPO version came slightly later, and we started with more traditional system. This vector, instead of TOPO version, it has just multiple clone inside. And my preference to use, instead of single promoter, I prefer to use dual P-fast-back vector because it allows you cloning of two different gene of interest onto two different promoters. And nobody forces you to put both. If you want, you can always put one. But if you are planning to work with protein-protein interaction, it's always better to have both genes at the same time. As a result, upon infection, each single infected cells will contain both of your target proteins, and there is always more chances to have this protein together for possible interaction versus if you will be using individual like recombinant viruses. In both cases, and in reality, I did not have any problem of finding respective restriction site. If you, let's say, your gene of interest has inside restriction site which is present in this cloning site, you can always try to use blunt end cloning or change the site. There is so many way around, but it's much cheaper than using TOPO version of the same kit. In both cases, if you clone your gene of interest under control either of P10 or polyhedron promoters, in both cases, sequence is flanked by poly-A sites. It's either SVW40 or TK poly-A. And it also has gentamicin selection marker for next recombination step. And the entire construct is flanked by T7 left and right sequences, basically creating mini T7 transposon, which is used in order to, at the next step, in order to create recombinant baculovirus. This cartoon depicts like overall schematic for recombinant baculovirus construction, and I will go briefly through each step. Basically, the first step, it's by either using P-fast-back TOPO or dual vector or regular vector. It's irrelevant at this point. You take your gene of interest and you clone in this vector. It's nothing is special about this cloning because it's regular cloning, nothing unusual. You create a recombinant molecule, you sequence, you confirm identity of your sequence, and after your sequence is confirmed, we go for the next step. It's transformation of competent DH10B back. Like it's very specialized cells. They Invitrogen offers these cells as like ready to go already competent cells. You don't have to do it by yourself. But if, for example, some problem with, let's say, it's very expensive again. But if you want, you can take these cells and make competent cells by yourself. Basically, you can do it by yourself, or you can just buy cells from the company. You transform this plasmid in this DH10B 10 back cells, and under selection of 3 antibodies, because these cells, they have backmid with kanamycin resistance, and it also has LAC-Z cassette for possible, like for blue-white selection. Donor plasmid has gentamicin, and helper plasmid containing transposase, which actually facilitate this transposition. has also tetracycline selection. And after adding all these three antibiotics, gentamicin, kanamycin, and tetracycline, and adding like special substrate, it's basically analog for like old version, it's X-Gal, they call it blue Gal, but it's definitely much better than X-Gal, and IPTG for induction. After approximately 24 to 36 hours, you're supposed to expect to see a very nice mix of blue and white colonies. And all you have to do, you just have to select those LAC-Z minus bacterial colony, which is supposed to contain your recombinant, like your gene of interest, already sitting in the right place of baculovirus genome, because this BACMID has like special site for binding, for T7 transposon binding, and because of homologous recombination, your gene of interest goes exactly in the right place of baculovirus genome. Now all you have to do, just pick up individual colony, and it's like one of possible problems if you can, I mean, you just don't have to mix those colonies. And as a result, in order to avoid possible mix-ups, they just recommend picking individual colonies, streaking one more time to make sure you have your recombinant, like your only recombinant construct and nothing else. And after, all you have to do, just do small DNA prep. It's basically like from 5 mil is more than enough for all this, because for real transfection in baculovirus cells, you don't need more than, like, you can use minimum 1 microgram DNA, you can use 2, 3, but this amount, like 5 mil culture will provide you more than enough DNA for all this, for all work. After, I forgot to mention that all these steps of initial transfection of infected cells and making like recombinant baculovirus stock through passages like P1, P2, or like P3 passage, typically they recommend to use adherent cell culture. And whenever, like from my experience, typically after passage 3, you have at least like 10 to the 8th virus particles per mil of infected cells. And after this passage 3, you are basically ready to go for pilot protein expression. And in this case, if you need just a little bit of, like not a lot of protein, or if you're not planning to do any purification, you can still go with adherent culture. But if you want to have protein for any possible future purification, it has to be suspension cells. The main advantage of these SF9 cells, which we are using in our lab, they can be easily adapted for cell culture, for suspension culture. Basically, if you have cells in suspension, if you need adherent, all you have to do, just take these cells, put on your plate, and after 15 minutes, they will become adherent cells, and you can use for transfection or making your virus stock. And you can, and because these cells, they do not require CO2, and they grow at like optimal temperature, 27 to 28, you can basically, I mean, it's relatively easy to adapt system. I mean, you can use a regular incubator, and for suspension culture, at least in our lab, we are using regular shaker, not even those special equipment, just regular flasks with RPM somewhere between 125 to 150, and it works just fine. It's and when I called companies, they even suggested to do this way instead of those pictures which Dr. Kasulas was showing before. This part now, it's like I will try to give some like I will try to explain some feature of this system by showing some result which we did receive in our lab from using baculovirus expression system. This particular results, it's they were published last year, and they were published in Journal of Virology. And in order to do, like for this publication, we had to produce a set of different recombinant baculoviruses expressing different portions of different herpes simplex glycoprotein. The main emphasis of this paper was like we it has been shown that amino terminus of HSV1 glycoprotein K does physically interact with HSV1 glycoprotein B. And in order to prove this, we had to, we did express the first 82 amino acid of glycoprotein K, and it was fused in a frame with 3X flag epitope because there is no antibody available to GK, and we needed some anti-flag antibodies. They are available, they're commercially available. So it's just for pure detection purposes. And like similarly, some other antigenic epitope anti-V5, like every single construct of GB contained anti-V5 epitope at the 3' end. And for this work, we used our dual vector system when at first we did clone GK on the polyhedron promoter, and those recombinant DNA later was used as backbone in order to put, let's say, GK plus GB154, GK plus GB364, again, GK plus GB500, and GK plus GB748. And basically, like the main conclusion from this slide, I can just say we have really relatively high enough level of expression for protein as low as it's in between 15 and 20 kilodalton, and we were successfully we successfully expressed different portion of GB ranging in size from approximately like 22 kilodalton to up to 100 kilodalton in size. So you can efficiently express small protein, and you can also efficiently express protein, at least in this case, as big as 100 kilodalton in size. Next, like unlike a previous slide, for detection purposes, we used very sensitive Western blot analysis and antibody. In this case, and those proteins, they were not purified at all. It was just straight cell lysates. In this case, this slide, we have a picture of protein gel, which was Kumasi stained, and all those bands here, they are real protein, and like for comparison purposes, line 7 shows BSA like marker with 0.5 microgram of DNA. It means like our best result, I mean, our best protein has at least like 2 to 3 microgram minimum of protein. And considering the fact that it's only small aliquot from the total protein amount, we can easily purify at least like from 10 to 15 microgram of protein from all these experiments. They were done from 50 mil of suspension culture. And I would like to point out that theoretically you can have two ways of producing protein in baculovirus infected cells. You can engineer your construct in such ways that protein will be either secreted outside of the cells. In this case, you're supposed to put typically honeybee melittin signal sequence in order to drive this protein outside of the cells. Or you can just use your sequence without any change, and in these cases, most likely protein will be cell associated. In this case, like all these experiments, they were done like 50 mil of cell culture of infected cells were used for protein purification. Cells were pelleted, and on this slide, supernatants were used for purification. Next one, I have the same cells, only cell lysates were used for isolation of the same GB154, 364, 748, and GD. It's also one of the HSV1 glycoprotein D. As you can see, we did not find any small protein GB154 in supernatant of infected cells. GB364 and GD like 340, they're very similar in size, and I mean, at least it's you cannot say you have very high yield, but at least we have very highly purified protein which can be used for some functional analysis. And the best the best yield was detected for the highest size of the protein of GB truncation. It's basically the entire soluble portion of GB gene was cloned in our P-fast-back dual vector. And if you look at next one for cell lysates, the same totally negative previous one, 154, was found in only in cell lysates. It means this small protein just gets stuck somewhere inside of the cells. It doesn't go outside. And GB360, basically we don't see any visible protein like for this GB truncation, and we have much smaller amount of the largest version of GB748, but surprisingly GD340, which is very similar to GB364, you have very high yield of highly purified protein. It means there is no guarantee, let's say, if you design your protein to be secreted or to be inside of the cells, you still have to try both ways, because you still can find your protein trapped inside of the cells. It means each individual protein require individual approach, I would say. It's not like you have the same rules like for all protein. And if you can look at a level of contamination, it's obviously then, for example, here we have much cleaner. We don't have basically any contamination, like contaminated protein for supernatants, versus we have like some across the board bands and like some other bands as well. In this situation, if you can, it's more preferably to use secretable version. But if for some reason your protein does not express as secretable, you still can go for the cells and try to purify your protein from cell lysates. Next one, we have some more example of using this system for some other protein. Let's say on lane 3, we have bovine spike coronavirus. It's the major antigenic determinant of bovine coronavirus. And this protein, like protein by itself, it's close to like 180 kilodalton, but here it was only soluble portion was cloned, and it's somewhere like at least minimum 120, 130 kilodalton. And another protein, it's basically the same spike glycoprotein fused with a bovine CD154 ligand, which is more close to 150. And as you can see, you definitely can tell that you can express protein as large as very close to 150 kilodalton. And we never had any problem with GB, because it's like, as an example, line 5 shows GB500. You have very nice expression for GB500. And the most interesting part is this protein, which is GK, and difference between this GK and our like previous one when we expressed 82 amino acid. In this case, we have the entire amino terminus of GK. It's like close to basically 117 amino acids. Level of expression is great, but if you look for left part, it's before purification, and right part, it's after. Let's say for GB, you have very nice enrichment, and you have very nice and clean protein after binding with specific resin. You still have some decent binding for spike glycoprotein, like not very well, but at least some binding for spike fused with CD154. But if you look at the entire amino terminus, there is nothing at all. It's just empty lane. And all this, I forgot to mention, all these purifications, they were done under native condition. It's, we did not use any denaturing agent at all. It's just under native purification condition. But it's only one obvious indication that most likely his tag, because his purification tag on this protein was located at 3' end of this construct, and most likely because of conformational changes, this tag sits somewhere inside, and because it's not accessible, it just does not bind to the column at all. So, oops, one second. Because this protein is very important to us, not because like its name KGK, because it stands for our boss Konstantin Gaskasulas, but it's because for the last 20 years, basically nobody was able to produce antibody against this protein. It's like we are working in at least in this lab for almost 20 years, and we still don't have antibody against this protein. So if we are able to purify this protein somehow and use for antibody production, at least it will be something which is totally new. So what we decided to do, like lane 2, it's our old version of the entire amino terminus of GK with his tag located at 3' end. And first we change tag location. Its tag was moved from 3' end to 5' end. And as you can see, basically you have very similar protein yield, only glycosylation pattern is slightly different. Like this is minimal molecular weight protein. This is glycosylated form. Here you have some kind of intermediate, but overall amount of protein is very similar. And as control, we have GB 500, which like we did not have any problem with this protein at all. And lane 5, it's the same 3' end labeled GK117. This time under denaturing condition. Denaturing condition means we used 6 molar guanidine chloride in all our solutions for purification. Still, you can see some traces of protein, like the same size, but basically binding is not very efficient. But at least for after location of the tag was changed to 5' end, at least we have some binding for GK118. We played some more with purification condition, like change some pH. And here we have, let's say, lane 2 and 3. It's 2 different samples, just straight cell lysates. And 4 and 5, it's the same samples, only after binding with resin. It basically says that almost all protein efficiently binds to the like to the resin under this condition. And line 6 and 7, it's protein after elution. So it looked like we found some suitable condition for protein purification, but again, at this point, it's still Western blot. I mean, it does not mean much until you go for actual protein purification by using Kumasi blue staining. Here we have 4 different, let's say, the lane 2 through 5, it's like four different samples of the same, basically. And in each case, we use 150 mils of infected cells from suspension culture. It's basically total, we have 600 mils of culture which was used for purification. And after only 1 round of binding with resin, we saw some, our expected protein is supposed to be in this area, and you can see some protein, but it's not clearly here, because this gel shows us some more picture. It's this picture after we took all those protein combined in once, and because it's contain 6 molar guanidine chloride, you're supposed to dissolve, concentrate, and after concentration, first of all, we have some precipitation because it's most likely too high concentration, but at least most of the protein still stays in solution without going in precipitate. And if you look at control BSA, which is the highest concentration, 1 microgram, we definitely have more than 1 microgram here. I would say like 2 or 3 microgram at least, maybe more. And considering the fact that I have like 100 times more of this protein than loaded here, I have at least, I would say, a couple hundred micrograms. So this amount is definitely enough to go for rabbit immunization. And basically, if we are talking about this amount of protein, at this point we may start switching from because all these experiments, they were done manually. Basically, you have resin. The main problem with resins, capability of each, let's say, for 1 mil of Talon resin from Clontech, which I use for this purification, can bind up to 3 milligrams of protein. You definitely do not have 3 milligrams of protein, and if you are using very big volumes, you cannot, because ratio between resin and volume of your protein solutions cannot be more than 1 to 500, maybe 1 to 100 maximum. But if you, let's say, if you have to use at least several milligrams of your resin, technically you cannot have even close this amount of your protein, and as a result, a lot of non-specific protein will be bound. And at this step, let's say you can do first round of purification, concentrate this, and after you can either use Profinia purification system, which we have, because at this point we have enough protein to go from manual to automated system, because all those cartridges on the machine, they have like minimum 1 mil of resin, and as a result, if you try to use at the initial step, purification step, you will waste your resin. And after some enrichment, you definitely can go, or maybe, I mean, you can find some other way of concentrating these proteins before going for automated purification system. It's basically, it's all what I have for the practical use of baculovirus expression.
Okay, thank you. We have a microphone here.
That's the mic.
That's the mic.
Although if you're facing the table, it picks up over here too.
Picks over here. Okay. So, oh no, you can hold it here for a second.
As long as it's,
You may have to answer some.
as long as it's on. You just want to stand so that you can both be
Right.
seen.
So, if you have any questions, we can answer them now. You can also email us any questions you may have. It doesn't sound, I mean, it sounds a little bit complicated. It's probably not as complicated as it sounds. And I always joke that in our lab, we could take an intelligent high school student and make him a biotechnologist probably in 6 months or less. Some of my graduate students probably would question that, but I think it's doable. So basically, you can get a smart high school student, undergraduate, and devote them to a specific project for cloning, production, purification, and I could do a really good job, especially if they have the guidance to do it. So there's nothing to be scared of, and after you learn it, you could do it multiple times for multiple different genes and proteins. So anybody has any question?
Start with Louisiana Tech. No? Any comments? Okay. LSU Shreveport?
None for me.
None for you. Anyone here? Don't worry, the camera's on them.
So we're going to put a request for training, or it'll probably go out early next week. We'll invite probably 2 or 3 people at a time to go through a specific module, either relating to a project that they need right now or even for training purposes. So we'd like to hear from you after we send this request for training out. You fill the specific information, send it to us, and then we'll pick different people, different campuses to come at different times to receive some training. And the training is not only on the protein production purification, but also on microscopy, as well as the other modules that we're going to be putting out, including FACS analysis, proteomics, X-ray crystallography, and other advanced methods later on. Part of what we're trying to do is to create a library of these training modules that are on tape or digitized to acquaint everybody, including students in your campuses. So I would like to see even undergraduate students that enroll perhaps in some of your classes out there to be able to log on to the LBRN website and view some of these technologies that they may use in the laboratory or they're assisting somebody doing it. So we can get a lot of these methods that we now take to be dispensed to not only to the faculty and graduate students, but also undergraduate students in your campuses. And we're going to be adding things. And if you have a specific idea or need in training in mind, and you want to hear somebody talk about different things, we'll invite core facilities throughout the state of Louisiana, from New Orleans, Pennington, to actually explain to us what do they have, how they use it, and how anybody else can access it. And perhaps after you know what is there, you have a better understanding what it is, it will be less intimidating, and you'll be able to approach them directly or approach them through us to be able to utilize the facilities and train. So that's what we're trying to do with all these modules. So hopefully you can help us disseminate that information to everybody. All right. Thank you very much.